Hello everyone, this is about the dark reactions which are also called as the biosynthetic pathways, light independent reactions, and also they are not dependent on light directly, they are dependent on the light products indirectly, and which is proposed by the Melvin Kelvin in the year 1950s also. And first what he has done is, during this World War time only, he was surprised and to see that why some plants are able to synthesize even under the normal conditions when carbon dioxide is more. So to find out that, he has taken a one jar, then in that jar he has kept the chlorella, which is a single cell algae. Then he used the isotopes, so that is carbon 14 attached to the CO2, then introduced into the jar. Then afterwards chlorella was found to prepare the photosynthesis. Then he killed the all the chlorella and everything. He has removed only the products. Then after removing this isolation of these products and everything, in that chloroplast he removed. Then in the inside the chloroplast he has removed some chemicals like A, B, C, and in that chemicals there is a pathways. He has seen that where carbon dioxide C14 is been traveled to the A and then B and then finally to the C. The product which is formed, he found out that it may be the sugar or it may be the sucrose or etc. Even in search of this only, Hatch and Slack, some other scientists also found out that even C4 also the same product has been released. By this analysis, he come to know that what are these three steps are nothing but carboxylation, reduction, and also regeneration, which is most commonest for every plant also. And in this only, he come to know that it occurs in the stroma, where generally in the plants, algae, and some of the bacteria also shows this, which enable carbon dioxide into converting into organic compounds like especially glucose. It is called as a dark reaction. Actually it is a misnomer, means wrong wrong thing, that it is not taking place in the night time. It do not require the light directly. So light independent Kelvin cycle also operates predominantly when sunlight is available, dependent on the light reaction products also. So simple reactions is RUBP in the presence of the enzyme. Most abundant and most commonest enzyme in the world only is Rubisco. Ribulose bisphosphate carboxylase oxygenase. When oxygen is there, it fixes oxygen. When oxygen is absent or low levels, then it fixes the carbon dioxide. So it is the only one which is having the two enzymes within one, carboxylase and also oxygenase. So it can fix the atmospheric carbon dioxide in the presence of this RUBP. Then 5 carbon plus 1 carbon becomes the PGA, then becomes the diphosphoglycerate, then becomes the phosphoglyceraldehyde. Phosphoglyceraldehyde, some of them will convert into again ribulose bisphosphate. So like carboxylation, reduction, and regeneration. Let's have a clear look at about this Kelvin cycle, where it start with so I have taken the 6 molecules like this, but generally we require the 6 cycles. 6 cycles for every time only one carbon dioxide is able to be fixed by the enzyme that is called it as an Rubisco. For our convenience, we are taking this all the 6 at the same time. 6 molecules of the RUBP, that is ribulose 1,5 bisphosphate, which is having the 1,5 bisphosphate. Phosphate is already having, it is a 5 carbon compound. So very important point, it is a 5 carbon compound, 6 molecules are involved. So then every time they are fixing the 1 carbon. So but here I have taken the 6 carbon dioxide. 6 RUBP, 6 carbon dioxide. 1 RUBP, 1 carbon dioxide. It will be as it is. So the fixing of atmospheric carbon dioxide in the presence of this Rubisco, this step is called as a carboxylation. So after the carboxylation step, the 6 carbon compound is forming. But actually this 6 carbon compound, 6 molecules are forming, which is called as a 3-keto 2-carboxy arabinitol 1,5 bisphosphate, which is a very short-lived intermediate. It is not staying for longer period, so it is splitting up. So it is splitting 6 molecules. If it splits up, how many we are getting? Actually we are getting the 12 molecules of the 3 carbon compound, 12 molecules of 3 carbon compound, that is we call as a 3-phosphoglycerate. 3-phosphoglycerate, in the presence of the phosphoglycerokinase by utilizing the 12 molecules every time 2 ATP, but we are taking the 6, so we are using this 12, and so we are using the 12 ATP. Then 12 into 1 comma 3 bisphosphoglycerate will be formed. Like this 12 molecules are forming 1 comma 3 phosphoglycerate. So in the presence of glyceraldehyde 3-phosphate dehydrogenase by utilizing 12 NADPH, by releasing the 12 NADP plus and also 12 inorganic phosphate, it is converting into 12 molecules of glyceraldehyde 3-phosphate. Out of the 12, 2 phosphoglyceraldehyde convert into glucose and also sugars, and so it will continue forming glyceraldehyde 3-phosphate 10 molecules. 10 molecules again further becoming the 5-carbon compound of the RUBP by utilizing the 6 ATP and also releasing 6 ADP plus inorganic phosphate, so that it is the process of is called it as an regeneration. It is called as an regeneration, and this process of preparation of the sugar we call it as an reduction. This is how these 3 steps are continued: carboxylation, reduction and regeneration. Again they combine, again the cyclic process will continue. So thank you for watching. Please do subscribe my channel. Thank you.